Hi everyone, have you ever asked a question about without the modification of your DNA coding sequence, how can a cell it can be differentiated to become your skin, to become your heart tissue? Actually, there's a lot of factor and study about that. This field called the epigenetic, and one of the typical example is about the methylation, which means on our DNA sequence there's gonna be this cytosine, just like a normal nucleotide, but it changed the hydrogen over here to become a methane over here, and we call it process is a methylatic. And if we have a lot of C methylatic C over here in near the promoter region, and it will prohibit the transcription factor bind to the gene, bind to the star of the gene, just like promoter region, and to start to make the transcription. Actually, if it's methylatic, it's very hard to it's will prohibit this process, and as a result it can cause methylatic gene expression is off over here. And the next question is about how can we know about this event. Just like from sequencing technology, the most thing that we need to know about bioinformatic technology, wet lab. And right here they just like add a special thing called the bisulfite conversion, which means from we can extract the DNA, later we add the bisulfite, and there's going to be special reaction over here. If the normal C cytosine, it will change into the uracil over here, and then later if we sequence them or make the B cell amplification, just like it will become the T. Instead, if the C is methylatic, and then after the bisulfite conversion there is no change at all, and then later for the sequencing or with amplification and sequencing later, it still become the C. So because of that, if we can get the sequence of the read, and we already know that it was treated with the bisulfite, so we can actually get the idea of how many of the methyl C are transferred. For example, right here, if we're sequencing them and we compare with the reference genome, very simple, just like calculate how many the blue point over here, and they are the C. It doesn't change anything when we add the bisulfite. And right here for the first position, just like we have 1/5, just like 20%. Similarly, we have 4/5, just like 80%. And because of that we can know like how much the at a specific position they are methylatic.